This video is going to be an introduction to cells. So you should know that all living things are made of cells or a single cell. So cells are considered the smallest unit of life. It's the smallest thing that can be considered to be alive. Now first, since we've already reviewed cell parts, or we should have already reviewed cell parts, I want to point out one important sort of way that some different cell parts work together. This is called the, I'll write it, secretory pathway. And what this is how cells that's job is to make those things and then transport them out of the cell. This is something that I've seen come up on the AP test pretty frequently, so if you know anything about organelles, this is a good thing to know. So there's lots of cells that's job it is to manufacture things, to make different proteins. For example, cells lining your stomach have to make digestive enzymes. Cells in your sinuses need to produce mucus. Cells in your nerves need to produce neurotransmitters. So there's a lot of different things that different cells produce. And the way that they do that, sort of the pathway that the things they produce take, is we would start at the nucleus, right? And the nucleus contains the DNA. From the nucleus, the process of actually making the protein from the DNA is something that we'll learn about later, but the protein is actually produced in the ribosomes, which are the little dots here, and usually that's going to be in ribosomes that are attached to the rough ER here. So the ribosomes will sort of spit the proteins into the rough ER. From the rough ER, the proteins will be transferred to a vesicle. It's just like a little transport bubble. From there, they're transported to the Golgi. The Golgi will modify them and then put them into transport vesicles to be transferred out of the cell. So this sort of pathway from nucleus with the DNA to the ribosome making the proteins to the proteins moving through the rough ER, potentially to a vesicle, to the Golgi, to a transport vesicle, and then out of the cell is known as the secretory pathway. So how those different organelles sort of work together to make that happen is, again, something that you might see come up. Okay. So next we're going to talk about the two different basic types of cells. The first type of cells are called prokaryotic cells. Prokaryotic cells are the earliest cell type to develop, so they're definitely the simplest cells. They consist of bacteria, which I'm sure you know bacteria, and also the domain archaea, which are similar to bacteria. They're single-celled organisms. They tend to live in more extreme environments, like in hot springs or areas that are really salty, but they're pretty similar to bacteria. The prokaryotic cells, they contain genetic information, so they contain DNA. The DNA of a prokaryotic cell is usually in sort of a circular chromosome, so a big loop, and then maybe some smaller bits of chromosome that are also little circles called plasmids, so a different sort of genetic setup to your cells or to an animal's cells. They also contain ribosomes because they need to be able to make proteins, and they have a cell membrane because there needs to be something to be the barrier between what is the cell and what is not the cell. Sometimes they will have a kind of a cell wall outside the membrane. They might have some little flagella or cilia if they're a cell that swims or moves around, and that's it. So all of those organelles that you learned about, the different cell parts, the Golgi, the endoplasmic reticulum, the nucleus, the mitochondria, All of those are not in prokaryotic cells. There are no true organelles, meaning organelles that have their own membrane inside of prokaryotic cells. There's just kind of DNA floating around in there with some ribosomes, and that's pretty much it. They also do not perform the process of cellular respiration, which is the way that food is converted into energy in, for example, your cells. They only perform the first part of that process, which is called glycolysis. And this is something we'll learn about in more detail later as well. But you can think of it as they have a much simpler metabolism, less efficient. They're not able to produce as much energy as a more complicated cell. The fact that they have no nucleus is kind of their defining feature. The word prokaryotic, the karyot part means like kernel, which kind of refers to the nucleus, and then this pro part means before. So it's like before there was a nucleus. So again, they've just got the DNA sort of floating around in there. It's not contained in any sort of internal membrane or a nucleus. The other basic cell type is the eukaryotic cell, where eu means true, and then karyot again means like kernel, but that can be translated as nucleus. So eukaryotic cells have a true nucleus, meaning they do have their DNA inside of a nucleus. They're also much bigger than prokaryotic cells. If you look at this picture, like this little mitochondria is more close to the size of a prokaryotic cell, and so you can see that the eukaryotic cell is much larger. Their chromosomes are linear, meaning instead of a big round circle chromosome, their chromosomes look like lines, or if they're duplicated, X's. So that might be more what you're thinking of chromosomes as looking at. Like, now, one way to remember this is that eukaryotic cells are you. You are made of eukaryotic cells, and so are plants, so are fungi, so are protozoas. So anything really that's not a bacteria or an archaea is a eukaryotic cell. So most of the things that you would think of as living are eukaryotic cells, again, except for those bacteria. They're defined by having those internal membranes, so they have all of those different organelles that we had learned about. They have the endoplasmic reticulum and the mitochondria and the Golgi and the lysosomes, the nucleus, of course, all those different cell parts. They have membrane-bound organelles that have their own membrane. So we say that they are compartmentalized. So if you think about like a junk drawer in your kitchen, right? The junk drawer is probably pretty messy if there's just all the stuff in there. But if you buy one of those like drawer organizers that has different sections, different compartments, you can put the paper clips in there and the pencils in there, and you can organize it. So eukaryotic cells are kind of organized. They're compartmentalized, so they can have certain processes and chemical reactions happening in this organelle and this part. They can have other processes and reactions happening in this part. And so they're able to be more efficient, really, than the prokaryotic cells because they have these different sections with these different membranes, where the prokaryotic cells just have everything floating around together like an unorganized junk drawer. So if you think about, like, why might it be useful for them to have these membranes, we have, again, that compartmentalization, so we can have different reactions happening in different sections. Another reason that it's useful to have internal membranes is to maximize surface area. So surface area is a concept that's really very important in biology. If you think about living things, you might notice there's a certain tendency to squiggliness, right? So like a brain, it's all squiggly, right? It's all folded and squiggled up. Your intestines, squiggly. a mitochondria. It's got squiggles inside. So the purpose of all of this squiggliness is to increase the amount of surface area, the amount of surface that is there, because if you were to take these intestines and like lay them out, they would be really super long and they wouldn't fit in your body. But when you squiggle them, you can get all of that surface area packed into a smaller space, a smaller volume. There is a disorder where people can be born with brains that are smooth and not squiggly, but since they have so much less area to think on, they're pretty severely mentally challenged because the lack of surface area. If you think about mitochondria, they're doing a bunch of chemical reactions to convert food into energy. And if you have an enzyme stuck here that can do a reaction, and one stuck here, and one stuck here, and one stuck here, and one stuck here on the squiggly membrane, you can fit a lot more of those enzymes than if it just had a smooth membrane. There's more area for you to stick these enzymes in if you have a squiggly membrane. So that's one reason that squiggliness is important. There's another reason that this surface area idea is important, so we're going to take a little aside and look at that. So let's think about cell size. Here's an elephant, right? Elephant, much larger than a human. Do you think that elephant has bigger cells than you, more cells than you, or both? So think about it, lock in your answer. So the answer is the elephant cells are the same size as your cells. They have more of them, they certainly have a lot more of them, but they do not have bigger cells because there's kind of a limit to how big cells can be. And the reason for that is that cells need to maintain that high surface area relative to their volume. So they want a high surface area to volume ratio. So let's make sure we understand what those terms mean, right? Surface area is how much surface something has. So for example, if you had a box and you wanted to know how much wrapping paper you needed to cover every surface of the box, that would be the box's surface area. Volume is how much space something takes up. So if you wanted to know the volume of your box, you could measure the length times the width times the height and figure out how much space is inside this box. So you can kind of think of the surface area as how much outsides you have, and the volume as how much insides you have. And so cells want to have a lot of outside area relative to how much inside area they have. And the main reason for that is that cells are always needing to get things in and get things out. In particular, most cells need to get oxygen in and they need to get carbon dioxide and other waste out. So if a cell's trying to get oxygen in and say it's this big, so it needs to get oxygen from here to here to get all the way inside the cell. And another cell is this big, and it needs to get oxygen from here to here to get all the way inside the cell. Well, this bigger cell, the oxygen has a lot further to travel to make it all the way inside the cell, which means it's going to take longer. If the cell is smaller, it's a lot quicker for the oxygen to get inside the cell. Also, if the cell is more squiggly shaped, like say like this, now the oxygen can go from here to here, and it's a much shorter distance for the oxygen to get. So the more surface area the cell has, the easier it is for stuff to get in and out. And the faster it is for stuff to get in and out. So what about size? Well, we can do some simple math to show that the bigger the cell gets, the lower its surface area to volume ratio gets, and the smaller a cell gets, the higher its surface area to volume ratio gets. And remember, they want a high surface area to volume ratio. So let's compare. These are obviously bigger than real cells, but a cube with 4 centimeter sides, a cube with 2 centimeter sides, and a cube with 1 centimeter sides as our sort of model cell. So if we do the math here, we can calculate the surface area of a cube by taking 6 times the side length squared, because there's 6 sides on a cube. And we can calculate the volume by taking the length times the width times the height, or the side cubed. So if we do the math, we get these numbers you see on the chart for the surface areas and volumes of our various cubes. So if you compare the surface area and the volume of each one, the surface area to volume ratio, or we divide the surface area by the volume, we find that the largest cube, let's see down here, has a surface area to volume ratio of 3 to 2, or if we divide it out, 1.5. And the smallest cube has a surface area to volume ratio of 6 to 1, or if we divide that out, 6. So this small cube has a much higher surface area to volume ratio than the large cube. This makes sense because as the cube gets bigger, while the surface area and the volume are both increasing, the surface area is being increased as the side squares, but the volume is being increased as the side cubes, meaning that the volume is increasing exponentially relative to the surface area. So the volume, the inside, is getting bigger much faster than the outside is getting bigger as we increase the size of the cell. This doesn't just apply to cubes. If we try the same sort of calculations with spheres, we're going to find the same results. So again, we can see that the smallest sphere that I did the math for has a surface area to volume ratio of 3, and the largest sphere has a surface area to volume ratio of 0.75. So again, the bigger the cell gets, the smaller the surface area to volume ratio gets. So this is why cells have to stay small. They have to maintain that high surface area to volume ratio, regardless of their shape. If you're comparing cells of the same shape, this rule will always hold true. So this picture is showing a microscope slide of inside the intestines. So inside of your intestines, the walls lining your intestines actually look like this, kind of like little fingers almost, squiggly. If you think about what the job of the intestines is, the intestines' job is to absorb. The small intestines absorb nutrients, and the large intestines absorb water. So since the job of the intestines is to move stuff through that membrane, it makes sense that inside they have this very high surface area to more efficiently transport those nutrients inside. In fact, if we look even closer, we would see that within these squiggles there are smaller squiggles. So it's almost like this. Squiggles upon squiggles. I'm saying squiggle a lot in this video, but I like the word squiggle, so that's fine. Squiggles upon squiggles, so really maximizing that surface area in order to be able to transport things through efficiently. All right, so that was our aside on surface area to volume ratio. Now back to the prokaryotic and eukaryotic cells. We know that the prokaryotic cells came first. The prokaryotic cells are much more simple, and they were the first cells to develop. So a question that has been on the mind of scientists is, how did we even get eukaryotic cells? How did eukaryotic cells develop? And the leading hypothesis to explain this, how we got eukaryotic cells, is called the endosymbiont hypothesis. So endo kind of sounds like enter, right, inside. And symbiont, you might know the term symbiosis, right, which is kind of like working together. So the idea with the endosymbiont hypothesis is there was a prokaryotic cell, and it tried to eat a smaller prokaryotic cell, nom. But for whatever reason, instead of eating that little cell, that it engulfed. It didn't actually break it down and digest it, but this little cell actually sort of went to work for this bigger cell. It became an internal symbiont, sort of working together. And so we think this is how the first eukaryotic cells developed. There was an event where something like this happened, and this little cell that got engulfed started going to work making energy for this cell, and that eventually developed into a mitochondria. And there was a separate event where another little cell got engulfed by a prokaryote, and that particular cell was able to harness energy from the sun, and that became a chloroplast. So we think, especially in the case of chloroplasts and mitochondria might have at one point back in history been their own cells, their own sort of like little bacteria that at one point went inside of another cell and just became an organelle for that cell. And those cells were more successful at producing and harnessing energy, and so they were able to reproduce more successfully, and eventually there were lots of these cells that had these internal symbionts, these chloroplasts and mitochondria. So this is just a picture sort of illustrating, again, that endosymbiont hypothesis, where the chloroplasts and the mitochondria were taken in by a host cell and then just became part of that cell. So this idea might sound a little crazy, like cells taking in other cells and then just making it part of itself, but there is evidence to support this. There's some good evidence to support this. One piece of evidence to support this is that mitochondria and chloroplasts are very similar to bacteria in a lot of ways. For example, they have their own separate DNA, separate from the host cell inside the mitochondria or the chloroplast. Another reason is that they have their own ribosomes, which are very similar to bacterial ribosomes, and they also make many proteins, which are very similar to proteins made by bacteria. So the idea that they are similar to bacteria supports the hypothesis that at one point, way back in evolutionary history, they were bacteria. Another piece of evidence to support this is that, again, similar to bacteria, mitochondria are made by dividing. So the body does not, or the cell does not manufacture new mitochondria from DNA. It doesn't put them together. Mitochondria make new mitochondria by splitting and dividing, just like how bacterial cells divide. In fact, you actually get all of your mitochondria from your mother. There's a sort of mitochondria starter set that's present in egg cells before they're fertilized, and then when that egg cell gets fertilized and develops into a baby, all of the mitochondria in that baby are just copies of the mitochondria of the mother that were present in that egg cell, and they just divide and divide and divide. You don't have any DNA to make mitochondria. If we took all the mitochondria out of your cell, you would be in pretty big trouble because you could not make more. The last idea I want to touch on with cells is cell specialization. When you think of cells, you probably think of that nice model cell, and it's like a circle, and it has a nucleus, and it has a Golgi, and it has an ER, and ribosomes, and mitochondria, etc. It has kind of all the cell parts in this nice round cell. But this model cell, while it's useful for us to think about cells, it's kind of a lie. Real cells don't really look like that. This is sort of like the prototype cell, like the everything cell, but actual cells that are within your body are very specialized. They have a special shape. Oftentimes that shape is not round. They have a special composition of different organelles and arrangement of those organelles for their unique function. Cells have jobs, and there's different jobs require different organelles, and they require different shapes. Like, for example, this cell right here is a nerve cell, and you can see that rather than being shaped like a circle, it's kind of this crazy branching star thing. So this is still very much a cell, but it's not, this cell. So one question that shows up on AP tests sometimes is about cell specialization, and it's what kind of cell would you expect to have these certain configurations or certain distribution of organelles? For example, what kind of cell would we expect to have lots of rough ER and Golgi apparatus? What job would a cell have that has that? Or how about a cell that doesn't even really have any organelles in there? No Golgis or mitochondrias or any of that. How about a cell with lots of mitochondria? So you can pause this, think for a moment, describe a cell that would sort of fit this description. What would its job be for each of those? These are 3 different cells. So if we're looking for a cell that has lots of rough ER and Golgi apparatus, my mind would go to that secretory pathway that we talked about at the beginning of this video. Rough ER and Golgi apparatus are both involved in secretion, especially the Golgi apparatus, because the Golgi are what package proteins into those little vesicle bubbles in order to secrete them out of the cell. So this cell that's being pictured here is a cell that is manufacturing mucus. So this might be a cell that's lining the inside of your nose or your throat, and it's producing these little bubbles, these little vesicle blobs full of mucus that it can then send out into your sinuses. So for that cell, I would pick a cell that's job is to secrete something. So the mucus producing cell, maybe a cell that is secreting hormones in the ovaries or testes, or secreting adrenaline in the adrenal glands. Maybe a cell that is producing and secreting enzymes, like one that's lining your stomach or some part of your digestive system. Okay. So this one might be kind of tricky. What kind of cell would be basically empty? No real organelles at all. The easiest one I think to think of is probably the red blood cell. The red blood cell's job is just to carry oxygen and carbon dioxide through the blood, distribute it through the body. And so these cells are really just empty little donuts that have proteins in there that are able to latch on and carry oxygen. So a red blood cell doesn't need to produce its own energy. It doesn't need to secrete anything or make any proteins or anything like that. So it doesn't need all that extra stuff in those organelles. It just needs to be able to transport oxygen around. Another example might be a root cell in a plant, which are basically just hollow tubes that water can move through, or a cell that's job is storage, like for example, a fat storing cell. It's basically just a sack full of fat. So lots of mitochondria. Mitochondria, you should think energy. Mitochondria's job is producing energy for the cell. So what kind of cell would need a lot of energy? Well, a muscle cell would be a good example, right? Muscles have to be able to move things and move your body, so that requires a lot of energy. Any cell that has cilia on it, so for example, there's cells in your lungs that have these little cilia hairs that wave around and beat and pump mucus sort of out of your lungs so that they don't get full of mucus. If those cilias are broken, you end up with cystic fibrosis, which is a terrible disease, or if you've broken them yourself by like years of smoking, then you end up with what they call smoker's cough, which is this gross hacking cough you get because your cilia are too damaged to move the mucus out of your lungs. But anyway, these cells, since they have to wave those little cilia around all the time, they need a lot of energy. Another cell type that needs a lot of energy is a nerve cell. It takes a lot of energy to send nerve impulses. So the cells in your brain actually use quite a bit of energy, and so they need to be furnished with energy from mitochondria. So those are a few examples that you might think of if you hear lots of mitochondria. So that's the end of this cell introduction. Of course, there are other types of cells that I haven't mentioned here, but just think about what the function of those organelles is, and then think about what kind of cell would need to use those organelles the most, and you should be able to come up with an appropriate cell.